Welcome everyone to this first endocrine grand round of 2024. We have the pleasure today to have Dr. Cypess as a speaker. Dr. Cypess graduated with highest honors in chemistry from Princeton University and then earned his PhD in biochemistry from Rockefeller University and then his MD from Cornell. He
he does these extraordinarily impressive 28, 48-day, internally domiciled studies where he knows every single thing a person ate. But we can't do that. And from what we saw, there was no change in the eating pattern. Interesting, though, we're studying a group of people now with polycystic ovary syndrome, and we've seen very altered dietary patterns. We had a person who had no change in her total mass or adipose tissue mass, and yet there were several days where total caloric consumption was 700 to 800 calories. And that wasn't a plan. So it led us to start looking at eating disorders within that group and wondering how it impacted, but that's not what we saw in this group of healthy women.
If you have questions, you can unmute yourself. Yes, Dr. Kidambi.
I have a couple of questions. Do you think the amount of browning, there's individual variability, right? So is there a certain threshold that for it to show some biological effect? So, and that's probably dose response thing. Is there any genetic impact? Obviously everyone is different. Is that playing, because when you get very small group of subjects, so maybe that's the reason why it doesn't show significant impact on weight. So.
Okay, so as before, these are really great questions that we think about. So is there a browning threshold? First, we don't know. So I can only, now that I say I don't know, I can then tell you what we think about it, which is that there doesn't seem to be an upper bound in how much brown fat someone could have. And that is suggested by the fact that there are some people with nearly a kilogram of what's detectable brown fat. So we presume that if we treated for longer, we would see more in these subjects, although we haven't done it. It could be that there's a plateau, but we don't know. And nobody knows yet if there is one. We don't know. There wasn't an obvious correlation in the trough levels of mirabegron and the amount of brown fat that grew. So we also don't know if that was what the impact was. The curious feature, I would say, beyond anything else to me, is that the brown adipose tissue depots that increased in mass the most were the ones in the inferior anatomical regions, the abdominal and the paraspinal, not so much in the supraclavicular region. And what I'm wondering about is whether that's because when we deliver mirabegron, it comes via the blood, and therefore there's greater blood flow to these regions, whereas when brown adipose tissue is stimulated physiologically, it comes through the nervous system, which seems to have more innervation in the superior regions. So that's one of the curious things that we had noticed in this particular study, but it's only 14 people. We are now doing a study of women with polycystic ovary syndrome, looking at between 10 to 16 women with placebo-controlled blinded studies. So it's a very rigorous study, but we haven't gotten through, we've done like 5 people so far, so we don't have a pattern yet to be able to share with you on that. In terms of genetic impact, we have the ability to look at the whole genome and whole exome sequencing in these people, but the N is quite small. However, I was telling, I'll get that question in a sec. I was talking, when I was at UT Southwestern, I was talking with Helen Hobbs about this because there are some people with way more brown adipose tissue than others. It is not distributed normally as far as we can tell. Is some sort of skew. So there may be some genetic factors that allow certain people to have much more than others. And it becomes an interesting question as to whether we'll have the informatics tools to find any of those genes based on the small N that we have. We can't, for example, just say, oh, that person has a lot of that. Bring in the mother, the father, and the siblings. You can't do PET CTs just on people like that unless you have the protocol in place to do that, and we do not. So we think that there's definitely genetic components. We don't know of any of them, and I'm not sure in mice whether there's been identified any that predicted, and I think that's because mice just have to have a lot of BAT. So whether there's a strain difference between 129s and black 6 or something like that, I don't know of anything at that level. But I will say, interestingly, that people have suggested that populations growing in warmer climates near the equator might have less than those who are in northern climates. What I know is that we've had several people in our study who came from Cameroon and other parts of Central Africa, and these young men had a lot of brown fat. So I don't think, it's not that if you come from an equatorial region, you won't have any brown fat. It's possible that people who live in Yakutia, there was a paper by a group now in Hunter, Stephanie Levy, where they used infrared imaging to suggest there might be more in people whose genetics really developed for a long time in really cold environments, but that has to be repeated. And so there might be enhancement in the polar regions, but not necessarily much issue with the equatorial regions.
Thank you.
There was a question in the.
Yes, there's a question in the chat. I will just read it for everyone, but the data that you shared support a critical role for brown adipose tissue or beige fat in energy homeostasis. And what is your opinion regarding the relative significance of adipose itself versus the central control of autonomic stimulation of the adipose tissue in the pathogenesis of metabolic disease? And what do you think about the relative utility of targeting central integrative control versus the adipose tissue itself to treat these diseases?
That's a great question. So let me just see here. Let's break it down. I typed it over so I can. Okay. The relative significance of adipose itself versus central control of autonomic stimulation of the adipose. I think that the driver is going to be the brain, and that the adipose tissue is more reacting than being the driver. But as you know, that you can have a driver in a car, and depending on the kind of car you're driving, you're going to have a different kind of performance. So that if one makes the adipose tissue more metabolically beneficial, then it can lead to good benefit. But in the end, it's going to. I think it begins with the brain, but the adipose tissue is important. But I think it's not the driver. At least that's what it would appear to be. Targeting central integrative control versus adipose tissue itself. Sure, it would appear to me, based on, for example, these studies with the GLP-1, glucagon, and GIP agonists, that the major impact is working on central control of energy intake when it comes to treating obesity. But getting a better a more healthy adipose tissue in and of itself could be beneficial, and it would very possibly be that the a more brown adipose tissue or better metabolically stated white adipose tissue could be helpful in responding to control from the brain, but also that the hormones released by these tissues might be beneficial for helping, like, for example, the heart to pump better, maybe making the bone a more metabolically helpful and more stable structure. And I sort of a point that I think is important to recognize is that mesoderm really talks to mesoderm a lot. And so it would be very logical, and I had a conversation with this today, actually, that I think that since brown adipose tissue needs good cardiac output, it wouldn't surprise me that the brown adipose tissue, when activated, is feeding back on the heart and helping it function better. And again, does it send a message to the brain to suppress appetite? I had a very fun conversation with Dr. Morselli about thinking about that if you have like one hormone that is orexigenic, but many more that are anorexigenic, there seems to be a drive for anorexigenic control at some point, and it would be logical that brown adipose tissue would tap into that during its energy expenditure phase. So I think that it starts with the brain, but the adipose tissue is important and valuable enough for metabolic health that it should require additional study and potential manipulation.
Dr. Mohammed, go ahead.
Fascinating talk. I have two questions.
I don't think we can hear you very well.
Can you hear me now?
Yes,
better.
So thanks for the fascinating talk. I have two questions. The first one, I think you came across very briefly when you answered one of the previous questions. If there is any, like associated, related epidemiological finding to this, whether that's like a ecological finding or observation or even gene enrichment that you find in a certain demographic, that's a little bit congruent or you find interesting related to the brown tissue study, whether that's in diabetes or weight or metabolism in general. And the second question, if you have any thoughts on implication of this especially in population with abnormal fat distribution like HIV and Cushing.
Okay, great questions. So the epidemiological findings, the kind that we've seen from, for example, Paul Cohen's study that I showed earlier suggests that having brown adipose tissue is associated with better cardiometabolic health. And our study and many others that were published 2 decades ago, and even in that time, have suggested that people with more detectable brown fat tend to be leaner and to have a better profile. But from the prospective studies where you're really activating the brown fat and you're seeing how much there most likely is, there hasn't been any obvious epidemiological information other than to say that people with obesity have less detectable brown fat. Now, whether that's because they truly have less or because it's not activated the same way because of issues related to glucose uptake and you have to use a different tracer and all, we haven't figured that out. So epidemiologically, it does seem that having more detectable brown fat is associated with a better cardiometabolic and general metabolic profile. In terms of populations with a normal fat distribution, Martin Toriani was a collaborator of mine from Mass General. He has looked at the abnormal fat distribution, for example, the buffalo hump in people who are taking antiretroviral therapy, and it had been postulated that there was brown fat that was in that region. We looked for it, and we published this paper, I think it was in JCE&M back in 2012, and we didn't see any brown fat in those regions, and I haven't seen any brown fat in the analogous dorsocervical hump that is in people with Cushing's and that classic Cushingoid appearance. For one thing, that buffalo hump region is not where you typically see brown fat. Brown fat's in a deeper depot, so it seems to be an abnormal white fat, but it requires more investigation to determine. But so far, no, it doesn't seem that these people with these very distinct, like for example, lipodystrophy, which we have a group that studies lipodystrophy here, people with partial or general, there was an attempt to use thyroid hormone to treat them, and there might have been an increase, but there wasn't a big signal in that area. So I think one of the things that goes in line with your question is we shouldn't think only of brown adipose tissue when we're talking about metabolic health. There are changes in the white adipose tissue, and there are many kinds of white adipose tissue, and these changes in white are also worth studying. We shouldn't think of it as WAT. We shouldn't think of it as subQ versus visceral. There's many different types. In Evan Rosen's paper, for example, that Atlas began to show that. So I think even if it's not that, I think that they're interesting different adipose tissue depots, and maybe in a new iteration of Netters, for example, we'll be able to say this is the adipose tissue depot of someone with Cushing. This is the adipose tissue with someone who's on antiretroviral therapy, and this is one with lipodystrophy, and this is someone with obesity. And the fat changes just the same way that the fiber types in the muscle modify based on exercise or other kinds of sarcopenic conditions. Oh, there's another question from, Dr. Kadambi, one more question. Any effect of beta blockers on BAT? Yes, there are. So in the original paper that we published in 2009, the New England Journal of Medicine, people who were prescribed beta blockers had much less detectable brown fat. And, for example, our colleagues at the Brigham and Women's Hospital were giving patients propranolol prior to their PET CT so as to suppress the activation of the brown adipose tissue. So beta blockers seem to be very effective at reducing brown adipose tissue thermogenesis. What proportion is central and what proportion is peripheral on the tissue itself is not known, based on the fact that these these beta blockers can bind to 1, 2, and 3. We suspect that there's a lot of peripheral intervention there.
Is that the reason, one of the reasons for weight gain with beta blockers? Usually, fatigue is considered as one of the, so is that?
It is a juicy hypothesis that brown fat might be contributing to part of the weight gain that we see with people taking beta blockers. And we posited that in the paper, but we haven't done any follow-up studies, and to my knowledge, nobody else has done either. But it would suggest that if someone starts a beta blocker for hypertension, if you could find a beta-3 agonist, for example, that isn't that doesn't lead to hypertension because it's at a right dose, or you've had another drug that could activate BAT thermogenesis, then you may be able to get around the weight loss, get around the weight gain problem that you see with people who take beta blockers, since they're so pervasive and for good reason.
Can I ask one last question? Sorry to keep you.
I'm here. I'm good.
Well, so what's the, so evolution usually, favors some of these beneficial traits. Why are no humans with super brown fat, like a heavy, like, I don't know, very large amount of brown fat? Because it seems it could be beneficial, right, as a human race. And so why isn't more brown fat in humans?
I've got an answer that might not be accurate, but it's simple. Because adult humans live in thermoneutrality. So it is possible that we evolved with a lot more brown fat than we currently have, because the caves, the houses were really cold. And so what we're seeing is an artificially reduced amount of brown fat in modern humans. And everybody here read Goodnight Moon, the kids' book?
Yes.
Goodnight Bowl of Mush, and the whole thing. Okay. So I looked at that. I mean, I read it as my mom read it to me as a kid, as a young child, and now I read it to my kids. But this time I read it as an adult. It's like, oh, that room is gonna be really cold. It's snowing outside, and there's a fireplace. But by the end of the book, the fireplace is out. I mean, this was like that was a fairly wealthy rabbit also. That was a nice room, right? So I wouldn't be surprised if you think about it that we used to have a lot more. And there was a study by Huttinen in 1981, I think, where they published the amount of brown fat they found in people exposed to the cold in Finland. And it wasn't a sophisticated, they didn't have really the genetic testing then, but there seemed to be a lot more. So one explanation that I give is that there may be way more brown fat in humans when we lived at a much colder environment, and that now we have artificially created a situation where there's less. That's the simple answer. It's an easy answer. It's attractive, but it may or may not be true.
I'd like to add that evolutionarily speaking, I don't know that it would have been a benefit to have a lot of brown fat from a weight and metabolic standpoint, because the issue that humans faced until recently was that there was not enough food, or the obesity was not an issue really. So that may also have prevented the development of super fat.
Yeah, that's true. That's one way to look at it.
Also, there's a lot in children, but we can't do the prospective studies in children. So I didn't even go into that, but the rate of detection of brown fat it was a paper I worked on with a group at Children's Hospital of Boston, where one could see maybe 50% of the children underwent PET scanning for clinical indications had detectable brown fat in a lot of it. So we also may be missing a very large proportion of the population who may have a lot more brown fat. And the way to do that is to hopefully get MR. I mean, Harry Hu and others have been working on using MRI-based techniques to be able to see, so that way you can study children. So it may be that it's in adults in particular that there's a lot less, and that there may be more in teenagers. But again, that's another study that would have to be done, and that's why we're pushing towards trying to use MR and MR spectroscopy-based techniques.
Thank you so much. Appreciate it.
Oh,
We have one last question.
Keep going. Let's go.
So you said that in your chronic mirabegron study, you found higher GIP levels.
Yes.
Can you speculate? Do you have an idea of why that is?
Oh, yes. I didn't show that. I think that there may be effects on the K cells that release GIP, and it may or may not be from the bile acid or other things going on. So let me see if I have I think we have, no. I think we had a model for this. I think it was either the adiponectin or the bile acids that were being made that then fed back to the K cells that caused GIP release. I apologize. I have developed a slide that we keep modifying, but we can plausibly connect activated brown fat, I think, either through adiponectin or through some of the bile acids that may feed on to the production of it. So that's what we think is happening. But we haven't done any studies to explain that. And before I mean, we passed the false discovery rate check. It certainly was significant in our group, but we got to confirm this again. Like if giving mirabegron causes an increase in incretins, that would be amazing. Is the level of incretin high enough to then have a suppression of the appetite that we're seeing with these meds? Probably not. These are physiological responses. So it may help as an incretin, but it may not have any effect on appetite suppression, which I would love to see. So that's why we're doing this next study.
Thank you. All right. Well, again, Dr. Cypess, it was a pleasure to meet you and to have you present on your research. Thank you very much.
Pleasure. And if any questions come up later, send me an email, aaron.cypess@nih.gov. Any question, anytime. Appreciate it.
Bye-bye. Thank you.
Thanks.